Communication between neurons and communication between neurons and muscle occurs at specialized junctions called synapses. The most common type of synapse is the chemical synapse. Here, we examine the events that take place at the neuromuscular junction, a chemical synapse using the neurotransmitter acetylcholine. Synaptic transmission begins when the action potential reaches the axon terminal. The resulting depolarization, due to the opening of voltage-gated sodium channels, initiates the sequence of events leading to the release of transmitter. Depolarization of the axon terminal membrane causes voltage-gated calcium channels to open, and calcium ions rush into the axon terminal. The calcium ions trigger the release of neurotransmitter by causing the synaptic vesicles closest to the active zone of the synapse to fuse with the presynaptic membrane. When the vesicles fuse with the membrane, they release their content of neurotransmitter into the synaptic cleft. The neurotransmitter moves across the cleft and binds to receptors on the postsynaptic membrane. The physiological response of the postsynaptic cell depends upon the particular neurotransmitter and receptor combination. In this example, acetylcholine binds to an ion channel, the channel opens, and sodium ions enter the postsynaptic cell. The signal is thus propagated to the postsynaptic cell. After a neurotransmitter has achieved its effect, it must be inactivated. For acetylcholine, an enzyme in the synaptic cleft, acetylcholinesterase, breaks down the acetylcholine into acetyl-CoA and choline. The release of transmitter from the receptors causes the channels to close. The choline is then taken back up into the synaptic terminal for resynthesis into acetylcholine. The synaptic vesicles are also recycled via endocytosis of the presynaptic membrane. The recycled vesicles are later refilled with neurotransmitter molecules and are ready for another round of synaptic transmission.